Hi, I'm David Hager from the Division of Pulmonary and Critical Care Medicine at Johns Hopkins University. Today's e-lecture will be on the control of ventilation. To start this discussion, I'd like you to consider a sleeping patient. As you know from your own personal experience, a healthy person breathes quite comfortably when asleep. Simply put, the brain, nerves, and muscles that control ventilation are on a sort of autopilot. You also know that when awake, you can control your breathing to some extent. For example, you can hold your breath and swim underwater. You can forcibly exhale to blow out birthday candles. Fortunately, however, the control of ventilation is usually in the autopilot mode. This allows you to listen to lectures like this without distraction. Anyway, back to the sleeping patient. There are a number of measurements we can make on the sleeping patient. With an electrode pinned to the diaphragm, we can monitor when it receives signals from the phrenic nerve to contract. You will remember that the esophagus is in the thorax and subject to the same pleural pressure changes that lead to lung inflation. If we measure the pressure changes in a balloon in the esophagus, we know how pleural pressure changes. If the patient breathes through a flow meter, also known as a pneumatochometer, we can measure the magnitude and direction of flow. Lastly, flow, when integrated over time, gives you volume. In short, there is an electrical impulse that stimulates the diaphragm. The diaphragm contracts, leading to a drop in pleural pressure, the lung expands, and gas flows into the lung. When the diaphragm relaxes, esophageal and pleural pressure rises and gas is expelled from the lungs. This sequence is simple. You will likely see similar displays of this data in your lecture on sleep-disordered breathing. In a normal person who should have a PaO2 of approximately 90 millimeters of mercury, the repeated sequence of events just described results in an alveolar ventilation designated by the abbreviation V sub A of approximately 5 liters per minute, as indicated by the red and green arrows, respectively. However, if you seal off the room in which your patient is breathing and drop the room's oxygen content, the patient's PaO2 will drop. The autopilot responds to the low oxygen tension by increasing alveolar ventilation, as shown here. The rise in alveolar ventilation is exponential when PaO2 falls below 60 millimeters of mercury. If you've actually ever done any climbing to any significant altitude, you may have experienced a similar effect yourself. As you know from your previous lectures, alveolar ventilation will correlate well with minute ventilation, which is respiratory rate times tidal volume. This is especially true in the healthy person. So, if you plotted minute ventilation against PaO2, the curve would have a very similar appearance to that shown here. The autopilot also responds to changes in CO2, as shown here. You will remember from your earlier lectures that a normal PaCO2 is approximately 40 millimeters of mercury. Even a small increase in PaCO2, as may happen when rebreathing into a paper bag, or due to an increase in CO2 production as occurs with exercise, will lead to a rapid increase in alveolar ventilation. The response to increased levels in PaCO2 is linear, as you can see. Again, as I mentioned in the previous slide, a rise in alveolar ventilation will necessarily mean a rise in minute ventilation. And if you were to plot minute ventilation against PaCO2, you would also create a linear curve, similar to that shown here. So why might these relationships be of interest to you? Well, the point is that normally, minute ventilation is very tightly regulated. As the prior two slides demonstrate, the ventilation autopilot adapts quickly to perturbations such as exercise, speech, altitude, and disease. If you observe a high or low minute ventilation, it tells you something about the patient in front of you. if you have an arterial blood gas and an estimation of minute ventilation, you actually know a lot about the patient. And if you're a pulmonologist, that's pretty cool. If you're a doctor of some other sort, this could be very useful. So here are some key points that we hope you take out of this lecture. Number one, have a general understanding of the interactions of the different components of the respiratory control system. Number two, know the anatomic location of the ventilatory pattern generator. Number three, understand the location and importance of chemoreceptors, both central and peripheral. And number four, learn to predict the ventilatory responses to hypoxia, hypercapnia, acidosis, and exercise. So this figure shows the general layout of the respiratory control system. It should emphasize that higher cortical function can override more rudimentary reflexes, such as the autopilot I've mentioned, also known as the pattern generator, which is located in the brainstem. As has been mentioned, holding your breath is a good example of this. However, if left to its own devices, the brainstem sends signals to the respiratory muscles telling them to flex and inhale, and then to relax, which would allow you to exhale. You then go forward, flexing and relaxing, inhaling and exhaling, etc. The output of the brainstem is modified by input from the airways, cardiovascular system, and even muscles and joints. For example, there are receptors in the lungs and thorax that are triggered when one reaches a large thoracic volume. These reflexes suppress subsequent respiratory drive, limiting further inspiratory effort or allowing expiration, depending on how you look at it. Perhaps more significant are the peripheral and central chemoreceptors that provide feedback to the brainstem as to the current state of gas exchange. As you recently saw, a rise in alveolar ventilation and thereby a rise in minute ventilation will follow a drop in PaO2 or a rise in PaCO2. We'll discuss this in more detail shortly. So this is the same slide I showed you previously. I'm showing it again to remind you that somewhere in the brainstem there is a quote-unquote pattern generator or pacemaker or autopilot, as I've referred to it, that leads to repeated cyclic stimulation of the diaphragm, breath after breath. So now I'm going to try to convince you of where this pattern generator or this stimulus is coming from. What you see here is a dorsal view of the brainstem. On the left side of the figure, you can see the general regions of the pons, the medulla, and the spinal cord. The horizontal dashes, numbered 1 through 4, are there to indicate where cuts will be made. On the right, you see a header labeled breathing patterns. So what you should look for is how the breathing pattern changes as the brainstem is transected at different levels. So, if the brainstem is transected above the PRG, or the pontine respiratory group, the breathing pattern looks pretty normal. There are 3 breaths of normal size, they're evenly spaced in time, and they're regular. When you look at the breathing pattern after transecting the brainstem at level 2, you should appreciate that the breaths are larger. They're still regular, but they're larger. This suggests that the pontine respiratory group may suppress the magnitude of tidal volume. In other words, the pontine respiratory group adjusts the size of tidal volume. When transected at level 3, the brainstem pattern becomes more erratic. There is rapid inspiration and rapid expiration. The interval between breaths varies. However, a robust signal is still being sent from the brainstem to the respiratory muscles. So now observe what happens if you transect the brainstem at level 4. What you see is a flat line. The respiratory muscles receive no signal at all. Breathing stops. This is what you get with a high cord lesion. You'll hear about that in your clinical rotations. So based on the series of slides above, it is therefore reasonable to conclude that the pattern generator is located in the medulla. Don't be confused by the name. The word pattern does suggest predictability, But I've just pointed out that if you transect the brainstem at level 3, breathing becomes erratic. The point that I'm trying to make is that breathing continues even after transecting at level 3, and breathing stops after transecting at level 4. So even though the pattern is irregular, its site of origin is in the medulla. What is lost when the brainstem is transected at level 1 or level 2 is what I would describe as fine-tuning. You can conduct the same series of experiments before and after transection of the vagus nerves. When this is done, breathing size and frequency is again modified. The implication here is that there is feedback to the brainstem via the vagal nerves. This feedback is interpreted and processed by the brainstem, which then modifies neural output to the respiratory muscles. There are a few other terms you may come across that I wanted to describe. The first is the pneumotaxic center, which is located between level 1 and level 2. This is also known as the pontine respiratory group, or PRG. The PRG has cell groups with inspiratory activity and others with expiratory activity. Logically, stimulation of these cell groups can lead to inspiration or allow expiration. The PRG also modifies the activity of the apneustic center by cyclic inhibition of inspiration. The apneustic center itself is located between level 2 and level 3. Apneustic breathing consists of prolonged inspiratory efforts with short expirations. In the previous slide, you saw that when there is a transection at level 2 and transection of the vagal nerves, there is apneustic breathing. So there must be feedback via the vagal nerves that is integrated by the apneustic center that leads to termination of inspiration. As has already been stated, the pattern generator itself lives between level 3 and level 4. So let's go back to the medulla and dissect a little bit more. If you were to stick electrodes into the medulla, you would be able to record the activity of neurons firing at different times during the respiratory cycle. Now, if you're dreaming of becoming a neurophysiologist with an academic focus on breathing patterns, please memorize this slide. If not, just know that there are a bunch of nerves that typically fire in sequence to drive inspiratory muscle contraction, then suppress contraction and allow expiration. Note that the density of the gradient in each bar reflect the intensity of firing of the related nerves. For example, early inspiratory neurons fire most intensely at the very beginning of inspiration, as shown in black, and then taper off to minimal activity, as shown in white, right before expiratory phase 1 begins. So this slide shows the muscle activity associated with pattern generation. In rows 1 and 2, you see a ramping up of activity during inspiration, followed by abatement of activity during expiration, which is usually passive. However, as you know, one can forcibly exhale, and in this circumstance, you could record an increase and decrease in expiratory muscle activity, as shown in row 3. There are two key groups of nuclei in the medulla that control breathing. Here you see the dorsal respiratory group circled in red, which lies in the nucleus tractus solitarius and is the origin site of inspiratory motor neurons. These neurons travel to the contralateral anterior horn cells to drive the diaphragm. It is also the projection site of visceral afferents via the cranial nerve 9 and 10. The ventral respiratory group shown here contains 3 nuclei with both inspiratory and expiratory activity as indicated. Importantly, it is the pre-Botchinger cells that contain the pacemaker for inspiration. In other words, the pre-Botchinger is the root site of the pattern generator, or the autopilot, as I have previously referred to it. All the other nuclei can be thought of as modifiers of this signal. After integration by the brainstem, the neural pathways that drive ventilation travel in the spinal cord. As shown here, voluntary inspiratory signals travel in the dorsolateral column. By contrast, the automatic rhythmic neural signals travel in the ventral and lateral columns of the cord. Okay, now take a deep breath and remember the following. Do not try to memorize all of the nuclei and neural pathways. You should have a general sense of where the respiratory pattern generator lives, i.e., in the medulla. You should know that the interactions of all the nuclei are integrated in the brainstem and result in output that drives the diaphragm and other respiratory muscles to a greater or lesser degree depending on what input the brainstem has recently received. To be more specific, here's what you need to know. The brainstem is important. The dorsal respiratory group has inspiratory activity. The ventral respiratory group has inspiratory and expiratory activity. The pattern generator lives in the medulla as part of the ventral respiratory group. The output of the pattern generator is modified by 1. The pontine respiratory group and apneustic center above, afferents to the brainstem from the periphery via cranial nerves 9 and 10, and other nuclei in the ventral respiratory group. Together, these components control ventilation. Okay, let's now change gears and talk about how the brainstem responds to changes in PaO2 and PaCO2. So we're going to talk about chemoreceptors. I showed this figure at the very beginning of the lecture. It shows that as PaO2 is decreased, there is an exponential rise in alveolar minute ventilation. This response is primarily mediated by peripheral chemoreceptors in the carotid body. The carotid body itself lives where the carotid artery bifurcates into the internal and external carotid artery. It is composed of 2 cell types conveniently referred to as glomus type 1 cells and glomus type 2 cells. Glomus type 1 cells generate output via cranial nerve 9 in response to low PaO2, high PaCO2, and low blood pH. Note that the response to hypoxia is determined by the PaO2 and not blood oxygen content. So, for example, an anemic patient with a normal PaO2 won't necessarily hyperventilate. Glomus type 2 cells don't really do too much. They're sort of structural support cells that provide a lattice work on which the glomus type 1 cells live. At the beginning of the lecture, we also looked at this figure, which shows a linear relationship between PaCO2 and alveolar ventilation. On the last slide, I told you that there is an increase in output from the glomus type 1 cells in response to high PaCO2. However, patients with surgically removed carotid bodies still have about 80 to 90% of the normal steady-state response to elevations in PaCO2, suggesting that most of the response to hypercarbia is driven by central chemoreceptors. It is likely that the peripheral chemoreceptors in the carotid body modulate breath-to-breath variation in tidal volume in response to changes in PaCO2, and that steady-state conditions are governed more by the central chemoreceptors. These central chemoreceptors are located on the ventrolateral surface of the medulla, which is bathed in CSF. They are also responsive to changes in pH, but this is a slower response because charged molecules don't travel across the blood-brain barrier as quickly as carbon dioxide. It turns out that the 2 ventilatory stimuli, hypoxia and hypercarbia, are not independent, but actually interact to amplify one another. Here we see the combined effects of low PaO2 and high PaCO2. The response to hypoxia is therefore amplified in this figure by elevations in PaCO2. Likewise, here, the effect of hypercarbia is amplified by hypoxemia. Recall that the hypoxia is primarily detected by the peripheral chemoreceptors in the carotid body. Again, we have an amplification of response. This figure reiterates that low pH increases minute ventilation. In the setting of respiratory acidosis, both central and peripheral chemoreceptors likely play a role. However, in the setting of a metabolic acidosis, the peripheral chemoreceptors will act first, and the central chemoreceptor will act more slowly to modify minute ventilation. This makes sense if you remember the following. Number 1, the CSF is separated from the blood by the blood-brain barrier, and number 2, it therefore takes longer for hydrogen ions to travel across the blood-brain barrier in contrast to PaCO2, which is rapidly diffusible. This is an instructive figure that demonstrates how many situations we observe in patients will alter minute ventilation. The curve in black is the one you've seen in the prior slides. Curve A shows the combined response of metabolic acidosis and rising PaCO2 on ventilation. The curve is shifted to the left. Curve B shows that during sleep, the normal response to elevated levels of PaCO2 is slightly blunted. The curve is shifted slightly to the right of normal. Curve C shows that narcotics further blunt the response. An extreme example of this is a heroin overdose, which we will certainly see in the coming year. Curve C also demonstrates the desensitization to chronically elevated levels of CO2 that occurs in some patients with COPD. Okay, so we've discussed the output of the brainstem, and we've also discussed chemical sensors that feed back to the medulla and modify its output. However, you should know that there are also sensors within the airways and the muscles of ventilation that transmit to the brainstem and modify subsequent output. These afferent signals are carried to the brainstem via either cranial nerves or spinal pathways. So I'm going to review some of the more commonly mentioned reflexes that affect breathing. The diving reflex is shown here. It's the body's response to submersion of the face in water. When this occurs, receptors in the nasal mucosa and on the face send afferent signals to the brain stem via the trigeminal nerve and suppress inspiratory efforts. It is this reflex that allows you to throw a baby into a pool. They generally don't inhale until you bring them back up to the surface. Obviously, it's the apnea part of this reflex that's key here. But in animals, a bradycardic and response and vasoconstriction have also been described. You may have heard about the Herring-Brewer reflex. It turns out that Herring and Brewer actually describe two reflexes. The more commonly known reflex is the inflation reflex. Here, slowly adapting stretch receptors are activated at high lung volume and send signal back to the brainstem, which leads to cessation of further inspiratory effort. Respiratory rate decreases and heart rate increases. With respect to the lung, overdistension is therefore prevented or limited. It turns out that this reflex is actually relatively weak in humans. However, if you take a guinea pig and give it one large breath, it will become apneic long enough to die. The less well-known Herring-Brewer reflex is the deflation reflex. In this case, rapidly acting receptors or irritant receptors are stimulated by lung deflation. The thought is that these receptors are activated when the lung is deflated abnormally, as is the case with atelectasis. Normally, we periodically take spontaneous deep inspirations, also known as sighs, which prevents significant atelectasis from occurring. This may be suppressed in hospitalized sedated patients who frequently develop atelectasis. Now that you know a little bit about what drives us to breathe and how ventilation is controlled, we will point out three abnormal patterns that you should be aware of in the clinical setting. The first is Cheyne-Stokes breathing. This is a crescendo-decrescendo pattern as shown in the figure. The intensity of the patient's respiratory efforts rise and fall so that they effectively cycle between periods of hyperventilation and periods of apnea. This is seen most commonly in the setting of brain injury and heart failure. Kussmaul breathing is characterized by deep, rapid breaths. In other words, the patient exhibits a very high minute ventilation. This is an appropriate response to a metabolic acidosis. As you will recall from earlier slides, the central and peripheral chemoreceptors are responsive to low pH. When the pH drops, minute ventilation goes up. So when you encounter a patient with diabetic ketoacidosis, remember this during your endocrinology lectures and on your medicine rotations, these patients may well be hyperventilating. Lastly, you've already seen the pattern of apneustic breathing in this lecture if you recall the series of slides where the brainstem was sectioned. From the earlier slides, you will also remember that if brain injury is associated with destruction of the brainstem or transection below the medulla, all respiration will stop. And this is clearly a very bad thing. Well, thank you for listening. I hope this lecture has been useful. Remember, our goal was for you to, number one, have a general understanding of the interactions of different of the different components of the respiratory control system. Number 2, know the anatomic location of the ventilatory pattern generator. Number 3, understand the location and importance of chemoreceptors, both central and peripheral. And number 4, learn to predict ventilatory responses to hypoxia, hypercarbia, acidosis, and exercise. Lastly, I wanted to make a comment that you should not underestimate the value of taking 30 seconds to watch your patients breathe. Based on what you have just learned, you will be able to think critically about what may be causing an abnormal ventilatory pattern. And this will be important irrespective of what subspecialty you choose. Thanks again for your time. Please don't hesitate to email me if you have questions. My email address is dhager1@jhmi.edu. Thanks again.